Okay, in this part we're going to talk about proteins, both the structure and function of proteins. So proteins are also polymers, just like carbohydrates, and the monomer that makes up proteins is called an amino acid. In this picture here there is a single amino acid. So on the right you can see in the circle there's a carboxyl group. If you remember, a carboxyl group contains a carbon double bonded to an oxygen and then single bonded to another oxygen which is connected to a hydrogen. And all amino acids have a carboxyl group as part of their structure, and that's what makes them an acid. And then on the left part of the picture there is an amino group, which is a nitrogen with two hydrogens attached. In between those two functional groups is a single carbon, which is connected to one hydrogen, and an R. Now, if you look on a periodic table, you're not going to find any element called R, and that's actually why we use an R there. R just means there's some other organic structure, and it can actually be quite simple. The simplest version of what R could be is simply another hydrogen attached there. And when that is a hydrogen attached, then that is the amino acid glycine, the simplest and smallest of all the amino acids. But there are actually 20 different versions of what R could be, and therefore there are 20 different amino acids. These same 20 amino acids are found in all living organisms on Earth. Every living thing on Earth is composed of proteins that are all just made up of different combinations of the same 20 amino acids. So that's pretty amazing. We're all pretty much alike in that regard. All life on Earth is very much related, and it could have been different. It could have been that all humans were made up of certain amino acids and all fungi made up of different, but that turns out not to be the case. All living things are made of these same 20 amino acids, so it's kind of like all living organisms on Earth are written in the same language. So, unlike the languages of humans, where there are many different kinds of alphabets and many different kinds of symbols that are used in those alphabets, in living things, these same 20 amino acids make up all the proteins of all living things. So I think that's very interesting. So we have a carboxyl group, an amino group, and a single carbon in between them. And this is the generic structure of all of the 20 amino acids. And it's only that R group that varies. Now I'm going to show you a chart of the other structures of the other 20 amino acids. And it's going to have the hydrogen removed right here, this hydrogen removed from the carboxyl group and actually added over here onto the amino group, and that's a way that these amino acids tend to exist in the cytoplasm. The carboxyl group will give up its hydrogen, which is what makes that an acid again, and the hydrogen will often stick to the nitrogen there, the amino group, because that is basic in nature. So just don't be surprised when you see that or alarmed by that. So here is a chart showing the 20 amino acids. Again, the simplest one, just a hydrogen in the upper left there, that's glycine. And notice that this chart divides them up based on their side chains. The side chains are what makes up the R group. They may be non-polar, such as the amino acids alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline, in addition to glycine. Or there may be polar side chains. That is, there's a little bit of a slight charge to the R group. And those examples are serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Or we may have acidic or basic side groups. So these are aspartic acid and glutamic acid in the lower left, and then we also have lysine, arginine, and histidine on the lower right. So these are all the 20 amino acids. But the interesting thing and the reason why they're organized this way in this chart is to show you that these side groups have different chemical characteristics. And these side groups then interact with each other when they are in a chain of amino acids, in other words, in a protein, by repelling or attracting each other, and this causes proteins to take on specific shapes, which is the most important thing about proteins. Whoops, went too far. So let's go back and just look at how amino acids can be linked together. As I told you in the video on condensation reactions, specific enzymes will recognize certain subunits and say, oh, hey, I know what to do when there's this amino acid and this amino acid next to each other. I take away the hydroxyl part of the carboxyl group, and I take away a hydrogen from the amino group of two neighboring amino acids and put those together. And that makes a water molecule. And then I connect up the carbon on the left to the nitrogen on the right. And now we have a dipeptide. A dipeptide is two amino acids joined together by a peptide bond. So a peptide bond is the bond that forms between the carbon of the carboxyl group of one amino acid and the nitrogen of another amino acid. And again, this is called condensation or dehydration. And in the opposite process called hydrolysis, different enzymes can break apart the amino acids by inserting the H and OH back on. So by inserting a water molecule and breaking the amino acids back apart from each other. So condensation and hydrolysis are the methods by which proteins are built and broken down. So when two amino acids are joined together, this is called a dipeptide. But when many amino acids are joined together, this is called a polypeptide. And a polypeptide is pretty much just another name for a protein. Proteins have very complex structures. That's the most important thing about them, is the structure or the shape that they take on. And the structure is completely determined by which amino acids there are and in which order. So looking here down this chain, this is the polypeptide chain that makes up this particular protein here. And the primary structure just refers to what are those amino acids and in what order do we have to link them together to make this protein. And then because of interactions of the side groups on each of those amino acids, the protein may take on a complex shape. If it kind of bends or kinks in a familiar way like this, that's called the secondary structure. It may be a helix or some pleated sheet. And then the whole protein, here's the pleats that we saw in the previous picture. The whole protein may fold up on itself. In fact, they pretty much always do, into some specific shape. And this is called its tertiary structure. And all proteins, pretty much all proteins, have what we call tertiary structure. That is, they are folded in on themselves in a very complex way. It's a complex way, but it's a specific way that is determined by the order of the amino acids because of the interactions of the side groups. And then if you have 4 polypeptide chains linked together, as we do in this case, this is the protein hemoglobin. And this protein is said to have quaternary structure because it consists of more than one polypeptide chain. So you could have a very complex protein that's made up of several polypeptide chains linked together, or maybe just 1 or 2 linked together. But as long as you have more than one polypeptide chain, that protein is said to exhibit quaternary structure. The functions of proteins are very diverse, but first and foremost you should know that they make up the structure of all plants and animals. So that's basically what we are made out of. When you're looking in a mirror, you're looking at all of the protein that makes you up. Your skin, your hair, your fingernails, your muscles and organs, everything about you is made out of protein. But also plants are made out of protein, fungi, bacteria, All living things primarily made out of protein, and that's probably the most important function of proteins, to make up the structure of living things. And thus you can see why the shape of a protein is so important, because you're made of the same 20 amino acids in different shapes to make up your proteins as every other living thing on Earth, and I'm sure you think of yourself as quite different from most other living things. That's because the shapes of your proteins are different. Proteins also make up the gatekeepers of the cell. In other words, they help regulate homeostasis by letting things into and out of cells. This is the phospholipid bilayer of the plasma membrane, and we'll look at this picture again when we talk about lipids, but think of this as the fence that goes around the outside of all cells, and inside the fence are little gates made of proteins, so those gates let things into and out of cells. I skipped a slide, so I'm going to go backwards. Proteins are what enzymes are made out of, and enzymes are so extremely important to living things. Without enzymes, almost no chemical reactions would take place inside cells, so we couldn't be alive. Enzymes are what make all chemical reactions happen, whether it's building up reactions or breaking down reactions. In other words, anabolic or catabolic reactions. All of these reactions take place because there is a specific enzyme that makes that reaction happen, that sees the two molecules, well, it doesn't see them, of course, but recognizes the two molecules and puts them together or breaks them apart. So besides making up the structure of all living things, serving as enzymes is the next probably most important thing about proteins. And then we did that one. And lastly, proteins are what most hormones are made out of. Now, not the sex hormones that we'll talk about in the lipids lecture, but most hormones are made of proteins such as insulin, and so they help regulate the processes, physiological processes in plants and animals. Plants also have hormones. And just another example, hemoglobin is a protein, and it's responsible for transporting oxygen to all of your cells. And there's a lot of other things that proteins do. So proteins are extremely important in biology. What they don't do is they don't provide energy for cells unless you have no other fuel source available. So cells do not want to burn up protein just to get out the energy stored in the bonds of the amino acids. That would be wasteful. That would kind of be like burning up your dining room furniture when there's a pile of firewood stacked up outside. So you're going to want to burn up the firewood first, and the analogy for a cell then would be burning up carbohydrates. But proteins could be burned up as fuel, but the cell prefers to use proteins to build up the structures that the cell needs. So that's all for proteins.